So let us see the structure of spermatozoan, mature spermatozoan. Okay, so that let me remind you that the process spermatogenesis. First is the spermatogenesis. Production of sperms is called spermatogenesis. During the process, spermatogonium into primary spermatocyte. Primary spermatocyte undergo meiosis 1 to form the secondary spermatocyte, and secondary spermatocyte by meiosis 2 it forms spermatids. The spermatid that develops into spermatozoa, we call the process as spermiogenesis. Then after that, the sperms which are embedded in the serotoli cells released out, we call the process as spermiation. Okay, just revising here that to understand much better. So finally, spermatozoon is formed. Okay. Then what is say the spermatozoon is a microscopic structure composed of three portions: head, neck, and middle piece. Then tail, head, neck, middle piece, and tail. Then a plasma membrane envelops the whole body of spermatozoon because it's just like a cell, isn't it? The head of spermatozoon contains an elongated haploid nucleus. Elongated haploid nucleus. The head of the sperm is covered by, here it is a cup-like structure that covered by a cap, a cup acrosome, we say it as acrosome. It is believed to be modified Golgi complex. You'll get this question. When you get this question, that is from cell biology it will become. Modified acrosome of the sperm is a modified Golgi complex. You need to understand. The acrosome contains numerous enzymes like proteases, acid phosphatases, hyaluronidase. These are the various types of enzymes that help the fertilization of the ovum, and penetration into the, let me remove this, penetration into the secondary oocyte or ovum. That will be helpful for that particular concept. Okay, so let's move there. That ovum, the middle piece we have to talk about. The head is over. The middle piece to be remembered is mitochondria. Numerous mitochondria are present in the middle piece, which produce energy for the movement of the tail. That sperm motility is essential for fertilization. Okay, so these sperms. So let me show you that the diagram. Let us see the diagram here. As we said, there is a head and neck. Neck portion is there, and middle piece which contain mitochondria which provide energy, and the tail which contain axoneme in the center. So this is the basic structure of this sperm: head, neck, middle piece, and tail. So this is the acrosome. Acrosome, which is a modified Golgi complex, contain various types of enzymes. Hyaluronidase, corona penetrating enzyme, the way that various enzymes are present there. So nucleus containing chromosome material with 23 chromosomes. Okay, sperms released from the seminiferous tubule, transported by our sequence we have, that accessory ducts, secretions of the epididymis, vas deferens, seminal vesicle, and prostate, essential for the maturation and motility of the sperm. Then the seminal plasma along with the sperm constitute the semen. As we said earlier, 60% is done by seminal vesicle and 15 to 20 or 30% by prostate gland makes the semen. The function of male sex accessory ducts and glands are maintained by testicular hormones or androgens. The sequence we have seen much better way in the earlier secretions of epididymis, vas deferens, seminal vesicle, prostate, that in order they have caput, corpus, cauda, epididymis, their secretions and secretions of vas deferens, seminal vesicle, prostate, they are essential for the maturation and motility of the sperm. One more thing here, the sperms, when they produce in the seminiferous tubules, once they produce in the seminiferous tubule, they don't stop, isn't it? They don't stop. They don't have any. They don't have any storage capacity. They have to go to the retitestes. Then from the retitestes, they move into the epididymis, caput, carpus, cauda. What all these are? Why this much longer ducts? You just imagine some 1,000 meter pipe. If you want to accommodate in the given space, in the 1 meter space, so how do you accommodate? You will rotate, you will rotate, you will rotate, isn't it? So that in that way we are talking about that our said seminal vesicles onwards, that everything for storage you have to say. You need to understand retitestis, vas efferens, vas deferens, that caput, carpus, cauda, everything is duct itself. Because there is no storage capacity. They have to move on. They have to move on. They'll move on like this. Okay. This is the fact. In the initially I said to you, I'll say in the end, this is the fact. Okay, there is no storage. When once produced over, they need to travel. Sperm need to travel. Sperm is continuously will travel. But in the mean travel itself is going to be the storage, you have to understand. Okay, along the length, that means you can understand the storage. That pipeline moving itself, the storage. Then after they go to the particular place. And let's see in the end the particular sequence here. Seminiferous tubule, retitestis, vas efferentia, that caput epididymis, carpus, cauda epididymis, vas deferens, along with the ducts of the seminal vesicle that will move into the ejaculatory duct by receiving the secretions of the prostate and Copper gland finally opens at the tip of the penis, urethra, we say. This is a complete sequence. Okay. So in the end, we need to discuss about the vigorous motility, shape, vigor, motility, shape, how do they play a vital role in the sperm. So these calculations we need to go here. A human male ejaculate 200 to 300 million. So 200 to 300 millions of sperms ejaculate during coitus, during copulation. At least 60% of these, at least 60% of these need to be normal shape and size. They need to have the normal shape. That means all the 200 to 300 million may not have, but at least 60% of them should have normal shape. So how much will find 200? 60% means 120, 200 to 300 we said. 120 to 180 millions of sperms should be with normal shape and size. Okay, at least 40% of them, that means in this 120 to 180, at least 40% of them should show vigorous motility for normal fertility. Okay, so normal shape and size, at least 120 to 180. In that 40% that need to show the vigorous motility, activate, very active. So how much it will come? 48 to 72 millions it will come. Okay, so 48 to 72 millions of sperm should have the normal shape, size, vigorous motility that will helpful for the normal fertility. Normal fertilization required 48 to 72. You may have seen over that in general wise outside we used to listen that 50 millions of sperms are required minimum for a normal count, sperm count. We used to say it as sperm count. In one coitus, that sperm count should be minimum 50 million. We just shown here a little bit technically 48 to 72. Okay. The calculations to be remembered. That means this also may be a question here. 200 to 300 millions of sperms is produced during the coitus. In that, at least 60% should be with normal shape and size. That means nearly 120 to 180. Then in that 40% need to be a vigorous motility, active, should be more active. So 48 to 72. Okay, so general terms we use it to say it as 50 million. Exact number, no need actually. But in examination point of view, if you see either 48 or 72 or 50, don't ask all the three if it comes what I have to put. So no one are innocent to give that kind of type here. So nearly number will come over 1 million, 40 million, then 100 million, 180 million will get. So nearly is 40 million. So we'll answer that. So in that way, okay. So that calculation, person to person, it may be different. So finally, this is the sperm and the structure of the sperm and the conclusion to the male reproductive system. Conclusion to the male reproductive system.